Good afternoon, my name is Dr. Stephanie McArdle. I'm the Senior Research Scientist in Tumor Immunology at the John Van Gys Cancer Research Centre, Nottingham, Trent University. And this afternoon I will be talking to you about some of our cancer vaccine that we think can be used potentially to prevent or delay the relapse of triple-negative breast cancer. Breast cancer is the most common malignancy in women, and TNBC, which occur primarily in young women under the age of 40, account for about 15 to 20% of all breast cancer. Compared with other subtypes of breast cancer, the survival time of TNBC is shorter, and the mortality rate is 40% within the first 5 years after diagnosis. And this is because TNBC is highly invasive and has a high early recurrence rate. Approximately 46% of TNBC patients have distant metastases at the time of diagnosis. And there are very limited targeted therapy available for TNBC because they do not express hormones receptors, nor do they express HER2/neu. Chemotherapy is the main systematic treatment, but the efficacy of conventional postoperative adjuvant with chemotherapy and radiotherapy is poor. The residual metastatic lesions eventually will lead to tumor recurrence, and the mortality rate of TNBC patients within 3 months after recurrence is as high as 75%. Immune checkpoint inhibitors have had a significant achievement across multiple malignancies, and the combination of anti-PD-L1 and Abraxane has been approved by the FDA with the frontline treatment of patients with metastatic PD-L1 positive TNBC. However, PD-L1 upregulation expression might be the result of cell transformation during tumorigenesis and therefore may not have the impact anticipated. Indeed, when researchers looked at the tissue sections of tumors, they were faced with broadly speaking, three scenarios. Either the tumor were not infiltrated at all with any immune cells, the so-called immune desert, or the immune cells were present in some clusters or at the margin of the tumor, or they were indeed heavily infiltrated. Now, depending on these three types of scenario, the type of vaccine that could be given, the type of treatment that could be immunotherapy treatment will differ considerably. Indeed, for a tumor that's not infiltrated, but removing the break by using anti-PD-L1 may not be efficient if there is no immune cells within the tumor. That is the case for when the tumor overexpresses PD-L1 as a result of tumorigenesis as opposed to immune pressure. And therefore, in this instance, one would need to first induce an immune response, and that can be achieved by many of these, and of which I will talk to you about vaccine, and more specifically peptide-based vaccine. And for this, we believe there are some criteria that would be required to have a good vaccine. The antigen itself needs to be chosen carefully. Indeed, one may not want to target an antigen that's expressed by healthy tissues, and especially if these tissues are vital organs. And you would need to obviously also choose which part of the antigen the vaccine will be targeting. The choice of delivery will also be of importance, whether the peptide will be used on its own or pulsed on top of dendritic cells or as an mRNA or DNA, and that will be important. If the peptide is to be used on its own, they will need, of course, some strong adjuvant because peptides are not immunogenic per se. And these adjuvant will differ in their immunogenicity. The route of delivery will also be important, and later on one may want to remove the bricks as well as or other immunosuppressive cells. So when it comes to the choice of antigen, Tumor antigen have been broadly categorized in two groups, the tumor-associated antigen or the tumor-specific antigen. Now, within the tumor-associated antigen, there are the overexpressed protein or differentiation antigen and the cancer-testis antigen. And I will talk to you about two protein that one is overexpressed and one belongs to the cancer-testis antigen. Ideally, one would target something that's unique to the tumor, such as the private neoantigen, because they are ideal in terms of tumor specificity. They would have no central tolerance and have a high prevalence in multiple patients. However, the private neoantigen, will be patient specific. So this is where it becomes more difficult. But for us, in our group, we've been targeting an overexpressed protein and a cancer-testis antigen. Again, a group by Shiver et al. Have published about the characteristic of an ideal cancer antigen. And these characteristics are depending on the therapeutic function. Indeed, if the tumor cells require the expression of this antigen, they are unlikely to downregulate its expression, so under immune pressure. Therefore, an ideal antigen would be needed by the cancer cells. The ideal antigen would be immunogenic, would be required for the oncogenicity. You would want the antigen to be expressed at high level and by positive stem cell expression. You would not want them to be expressed by the stem cells, right? Oh, cancer stem cell, yeah, but not stem cell. You would want a high number of patients to have their cancer positive for that antigen, have also multiple epitopes that could be used in the vaccine, and the cellular location of expression may also influence. So following these criteria, we believe that WT1, we're not the only one, WT1 is a good target for immunotherapy because it is expressed in various types of both hematological and solid cancer. It plays a role, oncogenic role, and it is immunogenic. This is just one paper quoting the in vivo vaccination of mice with WT1 peptide, which induce CTL, leading to tumor rejection, but there are other papers out there. Moreover, there is the ID and subcutaneous vaccination of patients with WT1 peptide 126, given with keyhole limpet hemocyanin in the combination with the subcu injection of GM-CSF, induced a reduction in blast cells followed by a durable complete remission. So, and in terms of breast cancer, WT1 was found to be expressed primarily by TNBC. So for TNBC, WT1 makes a very good target. It's currently in a clinical trial, phase 1 or phase 2, there are 3, one of which I think is finished. This just to highlight that the WT1 as a good target for immunotherapy. Following from the criteria of a good antigen, Shiver et al. Rank all the antigen that they have assessed, and WT1, with no surprise, comes at the top of the list. The one that we are interested in my group is NY-ESO1, TRP2, but also prostatic acid phosphatase and HAGE. Now, prostatic acid phosphatase, PAP, is not in this list because, if you remember in the criteria, one of the criteria was that the antigen had to be expressed by multiple cancers, whereas PAP is expressed only in prostate. It's still a very good antigen, but only for prostate cancer. As to HAGE, it's not yet in the list. Maybe not enough people have worked with that particular antigen, and that may change. As you can see, it's got 3 names. It's called helicase antigen, but it's also called DDX43 and CT13. This is because HAGE belongs to the cancer testis antigen, but it's one of those that's not expressed, not encoded in the X link, but it's a non-X link, it's chromosome 6. And it also belongs to the dead box protein. These proteins are called dead only because the core domain share the motif spelling the word dead. And it is an ATP-dependent RNA helicase, which helps to unwind the mRNA. And its name is DDX43. We have looked at HAGE expression, and we found it to be expressed in many different cancers, but not in their normal tissue match counterpart. But it is expressed by the testes, but not by vital organs such as brain, heart, and liver. So again, reiterated that HAGE is a potentially good candidate. When it comes to triple negative breast cancer, we found it to be expressed in 14.8% of early primary TNBC, but in 43% of locally advanced primary TNBC. We've also found that it is involved in promoting tumor proliferation via RAS signaling pathway, and therefore, again, we believe that HAGE is a good antigen. So when it comes to the choice of delivery, we have collaborating with Scancell, which have a platform called ImmunoBody. And these vaccines are a DNA-based vaccine that encode an immunoglobulin with no specificity, but where the CDR region has been replaced with the peptide sequence of the antigen of interest. So for us, we have the ImmunoBody HAGE, and we have the ImmunoBody WT1. When the ImmunoBody as a DNA is injected, for human is electroporation, for mice is in gene gun. When the entry is forced into the cells, the DNA then is transcribed into an mRNA, which then translates into a protein, and the protein being an immunoglobulin, it is secreted. Being secreted and having an Fc fragment, it is taken up by professional antigen presenting cells, which have got the Fc receptor for it. And inside the cells, the immunoglobulin is processed and presented to both MHC class 1 and MHC class 2, to CD8 and CD4 respectively. This particular vaccine, especially the one targeting TRP2 and GB100 called SCIB1, has already been used in a phase 1 clinical trial and proven to be safe and to generate cytotoxic T lymphocyte against the peptide that it carries. It is now in a phase 2 clinical and just entering a phase 2 clinical trial with the addition of anti-PD-1. To assess our vaccine, we are using the double knockout, double transgenic HHD2 DR1 mice, and we believe that these mice are a good model to assess the vaccine efficacy. These mice have been double knockout, so they do not express any of their murine MHC molecules. Instead, they express the chimeric HLA2 here, where the alpha-1 domain and alpha-2, which bind the peptide, are human. The alpha-3 with its transmembrane domain is actually murine. So this is very good because from the T cells, the CD8 can then bind to the alpha-3 domain. But the HLA-DR1 is completely human. Our immunization procedure that we followed for the ImmunoBody is 3 shots at 1 week interval. 7 days after the last immunization, the spleen are harvested and some form of functional assay is then performed. For the sequence, we've identified a 30-mer derived from HAGE. As you can see, this sequence here, it contains several peptides, 9, 10, or 15 amino acid, which have been predicted using the software freely available Syspathy to bind to several HLA haplotype. Here we've just mentioned HLA-A2 and DR1 because that's the mice we're using, but there are additional peptide capable of binding to DR4 and I believe HLA-A1. The binding score is interestingly quite high, and we have 3 peptides that are very close in their amino acid sequence, and that's worth remembering because in the next slide I will show you the result. We have a 2 9 amino acid long, but this sequence is shifted by 1 amino acid to the right, and that has had a profound effect on its ability to be recognized by T cells, which is interesting. And the decamer here is only 1 amino acid more than this peptide, and again, remember this because it has had a tremendous effect as well. The WT1 is 15 amino acid long, and it has inside 2 9 amino acid long predicted to bind to HLA-A2 molecules. So after the immunization, ELI spot was performed. So these are the number of spots per 500,000 splenocyte, and as you can see, peptide, which we call P4, did not respond, whereas the P5 and P6 gave significant response compared to cells alone, as did the P7, the 15-mer, but not the P8, the other 15-mer. So that just demonstrates that while the cyspathy is very good at predicted, it's not a guarantee, and one needs to assess using these mice. The WT1 has a very good response with the P3 peptide, to some extent to the P1, and not so much with the other peptide. So once we were able to demonstrate the immunogenicity of our vaccine, we then wanted to know whether these vaccine-induced T cells could recognize and kill target expressing these antigen, in order to demonstrate that the processing was similar when the whole protein was present within the cancer cells. So for this, we prepare some targets, and we prepare the 2 breast cancer, triple negative breast cancer cell line, the MDA231 and MDA468. Both express HAGE, but the MDA231 does not express, is HLA-A2 positive, sorry. The 468 is not. So that was a good control. We also needed, and both of them express WT1, the B16 cells, which have been knocked out for their own beta-2M, do not express any murine MHC. So these are of the same species as the mice that we're using, so they are C57 background. And they were then transfected with the chimeric HLA-A2, so the HHG2 and the DR1, and also further transfected with HAGE, and these express WT1. So here these confirm the fact that they do express HAGE. They also express the luciferin, which allow us to monitor the tumor growth using the in vivo imaging system. And they express HAGE, and here this just to demonstrate that they also express WT1. So when the mice were immunized and the spleen were harvested, they were splenocytes were re-stimulated in vitro with the peptide. This is to increase the number of HAGE and WT1 specific T cells in order for us to perform some killing assay. Here is to show you that mice immunized with HAGE were able to recognize specifically, more specifically, the 231, which is HLA-A2 positive, more so than the one that's negative for HLA-A2. Both express HAGE. On this side, this is to demonstrate the ability of the combined vaccine to recognize and kill the B16 cells expressing these antigen compared to the cells not expressing the antigen. Interestingly enough, when we immunize the mice with both HAGE and WT1, while the level of interferon gamma producing cells was identical to as if we immunize the mice only with HAGE or only with WT1, when we did some killing assay, we saw a significant increase in the ability of the cells to kill when the mice were immunized with both vaccine. We then wanted to know whether or not the vaccine was able to protect the mice either in a prophylactic setting or in a therapeutic setting against the growth of the B16 expressing these antigen. So for the prophylactic study, the mice first were immunized, 3 shots, and then they were challenged with a lethal dose of the B16 cell line. For the therapeutic, was the other way around. The mice were first given the tumor, and then the therapy started. And then the control group was neither immunized nor neither in prophylactic or therapeutic, so they received no vaccine. To the right here, you can see that mice that were pre-vaccinated, after they received the implantation of the B16, a significant decrease in the growth and the signal was seen as a total flux here with bioluminescence. And however, a group of mice can be seen to very quickly be resistant, if you want, or escape from the immune cells and grow. At this point in time, it would have been worth potentially immunizing or continuing to immunize the mice. Here, the therapeutic setting, the tumor held off a bit, but the mice eventually succumbed. You can see better here, whereby the mice that had been pre-vaccinated before the challenge were able to significantly delay the growth of the tumor and therefore the death of the mice. We are actually currently, our experimental design has changed, and we now pre-vaccinate the mice, challenge the mice, and then continue immunotherapy thereafter. The mice that were survive longer, there were 5 of them here, and the study was terminated before they needed to in order for us to monitor the immune response of these particular mice. And with their spleen, we did an ELISpot assay, and again we demonstrated that the T cells, the cells could respond to the HAGE and the WT1 peptide. But the mice that had been culled because the tumor was too big and they reached their endpoint, as you can see, we could detect no vaccine-specific T cells, or HAGE and WT1-specific T cells. Moreover, the splenocyte derived from these survivor mice, we could stimulate them in vitro with a peptide and demonstrate that we could get an enhanced response to the vaccine. So we had more interferon gamma expressing cells. Interestingly enough as well, these HHD2-DR1 mice have a tendency to be skewed towards a CD4, so the splenocyte have got a lot more CD4 than they have CD8 cells, but in the tumor it was the opposite. Primarily CD8 cells were found. But these CD8 T cells, while in the spleen they're not PD-1 positive, they were almost all positive for PD-1. Hence, we are now using the vaccine in combination with anti-PD-1. So in conclusion, I've just shown you that the DNA vaccine delivery ImmunoBody, which has got the advantage of both DNA and peptide vaccine, was able to generate some strong immune response against the peptide it carries and also delay their response towards delay the tumor growth. mainly prophylactic. The advantage of both adjuvant and delivery system are likely to be antigen and route of delivery dependent. That I didn't show, but I can tell you. In the future, we aim to remove the brake and or immunosuppressive cells to improve our vaccine. And we believe that the HHD2-DP4 double transgenic mice might be a better model. I would like to thank you. And on this note, the work was done for HAGE was done by Divya Nagarja, and the work on WT1 plus HAGE was done by Rukaya. Thank you very much for your time.